Morning, everybody. It's an honor for Quanterix to be presenting in this group. So Quanterix are really driven by sensitivity, and the reason is nicely illustrated by this chart, where you're looking at disease progression, and really where treatments are today is really on the right-hand side, where diseases are very progressed, especially in cancer and neurology, and we're looking at biopsies, at tumors, and often that's too late for patients, although these incredible treatments now are making it possible that they can be treated. And we think it's a lot to do with the ability to detect those diseases. And kind of related to that is the amount of molecules that you have associated with those tumors. You've got a high concentration. It's not so bad in cardiology and infectious disease, where this early detection is possible. Really what Quanterix is trying to do is to move that to the bottom left, where you're starting to detect molecules in healthy people, and this creates a lot of analytical challenges in terms of sensitivity. And the y-axis is really the sample that you're using to measure it. Currently, as we've heard, really it's biopsies, and in neurology it's spinal taps. And what we want to do is move things to low concentrations in easy to access samples like serum in particular. So that's kind of our general approach. And in terms of this community, really the hypothesis is, can we measure molecules, proteins in particular, that are part of the checkpoint inhibition pathway in serum? So in theory, this should work if we have the treatments that cause T-cell activation. That is characterized by an increase in cytokine concentrations, as we saw in the last presentation. And so it would be great to just go ahead and start to measure those cytokines in the periphery, in blood or in tumor cells, if you want to go that way. And this could offer a path to clinical companion diagnostics, as we've heard about more on the pathology side of things. This all sounds very nice, but you've got to overcome a technical challenge, which is, can you measure the molecules in the first place? And these molecules are circulating at the femtogram per mil sensitivities, which is about 1,000 times lower than what traditional immunoassays can detect. So you've got to figure that out. And that's kind of what our company has been doing for the last 12 years or so. And so we have instruments, we have assays, and we provide services. So I guess one big change since I last gave this kind of talk is we've added a second technology. We merged with a company that was just down the road, and now we have two platforms, one based on bead assays, one based on planar assays. So on the bead side, we have the HD1, and we just announced the HDX, which is a fully automated immunoassay system. The SRX has come out as well in between since the last presentation, and it's a benchtop version, a bit easier to use. And the SPX is the planar technology, which I'll get into. Both have kits. And then I think on the services side, there's been another change with our company in that we're now moving more towards diagnostics, and we're offering testing services, not just for clinical research, but also with a clear approval so we can start to test patient samples. So I'm going to go over the two technologies and just give some examples in the brief time that I have. But on the bead assay, which is where we started, really what we're doing is we digitize the detection of proteins. So in a traditional ELISA, you need millions of enzymes that are labeled with the proteins to generate enough signal to overcome the background in your assays to be detected. So what we did with Simoa was we isolated those proteins, enzyme-labeled proteins, in wells that are about 2 billion times smaller, so they're about 50 femtoliters. We seal them up, and if there's an enzyme in one of those tiny wells, it produces enough signal that's trapped that we can, in about 30 seconds, count that. So you go from needing millions of molecules to needing a single molecule to detect. We described this in Nature Biotech in 2010. It's 9 years, it's hard to believe. Things haven't really changed very much on the bead side. The way that we do this is we have a sample, and our favorite sample is blood, serum and plasma, but pretty much anything where you can get your molecules into a liquid that's particulate-free works very well with Simoa. And these beads are really efficient at going and grabbing all those proteins. We then form an immunocomplex on the beads with an enzyme label. We flow the beads into our Simoa disk. They fall into the 240,000 wells. We seal them up with oil, and then we start imaging those arrays, and we can determine whether there are beads in the wells and whether there's single enzymes associated with those. And so we can count single proteins in that way. So this has really taken off in the area of neurology, and cancer has been an area that we're seeing more impact. And I've just got a couple of examples here, especially in the immuno-oncology area. I would say our partner, Myriad RBM, who do a lot of testing services, they have really pioneered this area. We don't see a lot of it because it's kind of confidential stuff, but they did have one poster that was a couple of years ago, and this is looking at interferon gamma, and it was a biotech startup in the UK who were looking at pre and post treatment and looking at interferon gamma levels, circulating interferon gamma levels, and they're able to see a pretty significant difference between before and after treatment. And that was really driven by the really high sensitivity of the assay. So you can see down there in the bottom left, it's 19 femtograms per mil. And we saw in the last presentation where you're having to use cytometry to measure interferon gamma in cells. Well, this is a way to do that with a simple blood test. We talked in the last presentation about how do you scale these technologies and make them available. If you have a simple blood test, then that opportunity is obviously much greater. More recent publication, this is Vanita Gupta from Genentech, and they did a really nice kind of analytical validation of a Simoa assay for IL-12. And this is a cytokine that is in the pathway and in theory is upregulated after treatment. And this is with an anti-PD-L1 inhibitor that was mentioned in Christine's presentation. And so they did a really nice analytical workup at this. They were looking at the levels in the blood of several different cancer types. And you can see, if you look at the y-axis, you can see the concentrations that we're having to measure. They're well below kind of picogram per mil levels. We're at the femtogram per mil levels, and we're able to quantify these in all the cancers. And the results before and after treatment were pretty interesting. It's a pretty small cohort, 16 patients, 12 of which did in fact have an increase in IL-12 concentrations, as you'd expect from this drug. 2 people didn't change, and then 2 people actually went up. So again, very early stages in terms of the clinical demonstration, but the analytical performance is there to make these kind of measurements. So in terms of kind of where this matches up to what's out there already, again, this is data from Myriad RBM, and this is a company that's been running Luminex assays, another bead technology, for more than 20 years. And the problem they run up into is limited detection. So if you look at the right-hand side, you're looking at, picogram per mils, tens of picograms per mils. You couldn't measure those patients with that technology. So the second technology they brought into their company was Simoa assays, and they're able to demonstrate, 1,000-fold improvement in sensitivity. And so in their studies now, I think they basically run Luminex and Simoa in all of their studies. So that's Simoa beads, and where we've found that most useful is really the low number of cytokines. We have 3-plex, 4-plex, that's kind of the sweet spot for the beads. We developed a second technology that we call Simoa planar, even though it's not really some single molecule just yet. It was a technology that we acquired when we merged with Ocean Biosystems. And it's a technology that's been around for 20 years, and it's pretty kind of, you'd have thought it was standard technology, but we've managed to what we call Simoatize it and to extract every piece of sensitivity we can out of it, and we're going to publish a paper soon on how we did this. But what the folks at Ocean do is it's a spotted array ELISA in a 96 well plate. So they can spot right now 12 different antibodies. You add your sample, they capture the proteins, they get labeled with another enzyme, this time HRP, and you add luminol, and those spots chemiluminesce. The really cool thing about the technology is that the imager that we use, and there's one just at our booth, takes a picture of the entire 96 well plate in one shot. So this means your read times are incredibly quickly. Instead of having to go read an image each well one at a time, you can take one picture. So we read 96 wells in just a matter of seconds. So we get asked, well, you said it was beads and single molecule, how is it working when you're using chemiluminescence? It's really the same principle. Our goal at Quanterix, when I look at it, is how do we get the maximum signal to noise for an enzyme? And with the planar technology, we've kind of optimized pretty much every single aspect of that to get a signal to noise that's almost comparable to the bead technology. So the way that we've done this, they have a really nice spotting technology, so they use pins that basically very gently spot the antibodies onto the plastic. Plastic's proprietary. They had a clever idea of spotting the arrays in circles, and this really helps signal consistency. The old-fashioned rectangular grids, you get this concentration variation across the bottom of the well, but if all the arrays are the same distance from the middle of the well, you get the same signal. So it's really about CVs, which is just as important for sensitivity as anything else. They image through the bottom of the plate, so you're not looking through a meniscus as you often do with other technologies. That helps signal to noise. And I'll give one example, but we spend a lot of time looking at interferences. So when you get to these high plexes, heterophilic antibodies can completely ruin an assay, and we put a lot of effort into that. And then in terms of analysis, so we are able to, we've got these very high density images, much like in pathology actually, and we use the same kind of astrophysics approaches to detect molecules in this technology. So this is a 10-plex, we call it, it's a human cytokine array, and Purvish, one of our employees, has got a poster after this, so go along and maybe even vote for him. He's a good guy, he would love to win the best poster award, I'm sure. But this is a 10-plex that Ocean had, and we kind of Simoatized it, and we really had to work on that interferences, so we had to come up with a buffer that allowed you to measure 10 different cytokines in these patients who may have a bunch of different antibodies floating around in their blood. And it's kind of 10 of the most common cytokines that are important in immuno-oncology. And we got LODs all in the femtogram per mil range. The poster goes into this. We're looking at precision and spike recovery, but it's a really nice, robust assay when you want to look at all these different proteins in one sample. We've just started to do kind of analytical validation, and our customers will be the ones that will be looking more at the clinical side of things. But we're able to look at healthy individuals' serum and plasma, and measure all 10 different cytokines simultaneously. The red lines are the LOQs, so for the majority of the patients, for the majority of healthy individuals, and for most of the cytokines, we can measure these with good precision. The data on the right is looking at healthy individuals and 2 different types of cancer. So you can see that the kind of the ability to get baseline information on these cytokines as people progressing in the cancer or receiving treatments, that's really where our technology can help a lot. So we're hoping that this can become part of the arsenal for oncologists in measuring serum biomarkers as well as the pathology that we heard about a lot this morning. So I think I've squeezed everything in. I do want to finish just on a slide. We often get asked, what is the importance of sensitivity? And so I put this slide together recently because I was thinking of all the reasons why people use sensitivity and how it's useful. Because it's not always obvious. You can say early detection of cancer, but that's really just one part of it. The way people have been using the technology is, yes, early detection, and there was a paper in Nature this year looking at a molecule, LIF, in prostate cancer, which looked particularly interesting, and they used Simoa. But really it's other things like, first of all, profiling healthy individuals to understand a continuum from healthy to disease. That often isn't possible with current analytical methods. Maybe the most important thing is less invasive testing. So we're able to measure things in blood or saliva or urine, things that are accessible where you can't always get to those tissues, and urology is the best example of that. You can't get to the brain, but we're able to measure those molecules in blood. Smaller sample volumes, this can be very important for researchers who are looking at mice or have very small amounts of sample that they've got to put across all these different technologies that want them. If you have sensitivity, then you can take a small volume, dilute it up, and get the result. We've shown examples of measuring concentrations with therapies. Therapies often reduce the concentration of proteins, which put them below the analytical detection limit, and we're able to do that with Simoa. You can use it for speed. So instead of, you can use your sensitivity to do a very rapid test, and we can do our tests in about 15 minutes in the case of troponin. And measure things in things that are hard to measure. So we've got a test for C. difficile that we've measured in stool, where we basically diluted stool 20-fold, filtered it, and we were counting single molecules of C. diff and showing that it's a much more specific test than PCR. So with that, I just thank you for your attention. I'm happy to take any questions or chat afterwards, and would love to see you with the poster.